Cell death is vital for maintaining tissue homeostasis, development, and defense against disease. It eliminates damaged, infected, or unnecessary cells, ensuring proper functioning and survival. The human body constantly balances cell generation and elimination to maintain tissue and organ integrity. Cell death occurs through necrosis and apoptosis. Necrosis is passive and uncontrolled, often triggered by injury or toxins, leading to inflammation. Apoptosis is active and programmed, crucial for development and immune regulation. Understanding these mechanisms is key to developing effective therapeutic strategies. Necrosis, or accidental cell death, occurs under harsh conditions disrupting the cell's environment. Unlike apoptosis, necrosis is uncontrolled and energy independent. It is triggered by external factors like injury, toxins, or extreme temperatures, leading to cell lysis. The plasma membrane's disruption causes water and ions to flow into the cell, leading to swelling and rupture. Lysosomal rupture releases digestive enzymes, breaking down cellular components. Necrosis often triggers inflammation. Apoptosis, or programmed cell death, is essential for homeostasis and defense against disease. This process is a vital component of various biological functions, ensuring that damage from acute cellular injury, inflammation, and damage to surrounding tissues. Apoptosis can be initiated by internal signals, such as DNA damage, or external signals, like death ligands binding to cell receptors. These signals trigger a cascade of events within the cell. One of the key components in this process is the activation of caspases, a family of protease enzymes. Caspases act as the executioners of apoptosis, breaking down cellular components in a precise and orderly manner. During apoptosis, the cell's DNA is fragmented, and the cell itself shrinks and condenses, forming small membrane-bound vesicles known as apoptotic bodies. These apoptotic bodies display specific "eat me" signals on their surface, which are recognized by phagocytes, the cells responsible for engulfing and digesting cellular debris. Phagocytes then engulf and digest these apoptotic bodies, effectively clearing them from the tissue. This process prevents the release of potentially harmful substances through this meticulous process and ensuring the health and no longer needed or are functioning improperly without causing harm to the surrounding cells. Distinguishing necrosis from apoptosis. Necrosis and apoptosis are distinct modes of cell death with unique characteristics. Is essential for medical research and treatment toxins. This leads to cell swelling, membrane rupture, and the eventual death of the cell, further tissue damage, and attracting immune cells to the site of injury, like certain cytokines. During apoptosis, cells undergo shrinkage. Apoptotic bodies are then engulfed by phagocytes, specialized cells that clear away cell debris. This process prevents inflammation and allows for tissue homeostasis. Key differences between necrosis and apoptosis include their initiation mechanisms, energy requirements, regulation processes, effects on disease. By knowing how cells die, researchers can design cancer therapy, where inducing controlled cell death in cancer cells can lead to significant medical breakthroughs and improved patient outcomes. Necrosis involves a complex interplay of molecular events leading to cellular disruption. It is often triggered by overwhelming stress, impairing mitochondrial function and ATP production. ATP depletion disrupts ion pumps, causing ionic imbalance and cellular swelling. Reactive oxygen species, ROS, accumulate, damaging cellular components. ROS increase Section 6. Molecular mechanisms of apoptosis is a regulated process essential for tissue homeostasis and development. It can be initiated through intrinsic or extrinsic pathways, both activating caspases. The intrinsic pathway involves mitochondrial release of cytochrome C, forming the apoptosome activates caspase 9, leading to execution or caspase activation. The extrinsic pathway involves death ligands binding to death receptors, activating caspase 8. Executioner caspases cleave cellular substrates, leading to cell dismantling. Morphological changes include cell shrinkage, nuclear condensation, and antibody formation. This controlled process prevents harm to neighboring cells. Section 7. Clinical significance and therapeutic implications. Understanding necrosis and apoptosis has profound clinical implications. These processes are fundamental to many diseases and conditions, making them critical areas of study in medical research. Necrosis contributes to tissue damage in conditions like myocardial infarction and stroke. When cells die uncontrollably, they release harmful substances that exacerbate injury and inflammation. Apoptosis, when dysregulated, is implicated in neurodegenerative disorders and cancer. Regulation of cell death is essential for maintaining healthy tissues and organs. Excessive apoptosis leads to neuron loss in diseases like Alzheimer's. This loss of neurons results in the cognitive decline and memory loss characteristic of the disease. This unchecked growth is a hallmark of cancer, making it a challenging disease to treat. Targeting these processes offers therapeutic potential. By understanding and manipulating cell death pathways, researchers can develop new treatments for a variety of diseases. Inhibiting necrosis could limit tissue damage in conditions like stroke. This approach aims to preserve as much healthy tissue as possible, improving patient outcomes. Inducing apoptosis in cancer cells is a major therapeutic goal. By triggering programmed cell death, therapies can selectively target and eliminate cancerous cells. Emerging therapies exploit apoptotic pathways to selectively kill. These innovative treatments hold promise for more effective and less toxic cancer therapy.